Bismillah Rahman Rahim. Today we'll talk about the second part of biofortification of staple crops. In the last lecture, we talked about staple food crops and their distribution worldwide. Then we talk about the biofortification of staple crops, the significance of biofortification of staple crops, especially with reference to developing countries. Then we talked about zinc and iron biofortification of wheat and rice through agronomy. So today we'll talk about wheat and rice biofortification with zinc uh, and iron in a genetic way. So we have a great potential of cereals like rice and wheat for zinc and iron biofortification. The reason is that we have a cereals which have inherently low concentration of zinc and iron in grain, and particularly when they are grown in the micronutrient deficient soils. And secondly, uh, currently about 50% of the global cereal growing region are soils which are low in zinc level and even other micronutrients as well. So it gives us great potential for the biofortification of cereals. Another important thing which is also beneficial when we talk about when we work about the biofortification of cereals, that the deficient concentration in the plant that is 15 to 20, then sufficient is 20 to 100 milligram per kg or ppm, and the toxic level that is much higher, and that is more than 100 milligram per kg. So it means even we have a capacity to enhance the concentration from above the 100, because there will be no toxicity, but we will get benefit by adding some zinc to the grains. So this is also a positive point when we go for biofortification or genetic biofortification of the cereals. The targets which has been achieved yet they are the wheat. The baseline was 25 ppm, and then the concentration achieved is 37 ppm. And regarding iron, that is 25 ppm baseline, and then the final target achieved is that is 33. In rice, it's a bit lower. Is the target achieved that is 28 ppm for zinc and 13 for iron. So, but still the people they are working on that because we cannot increase the concentration in a single year, as we have discussed in the previous lectures. So we have to enhance it gradually. So the people are working on that, and hopefully they will get even more concentration in the grain. When we talk about the genetic biofortification of wheat, the existence of genetic variation within the plant genetic resources by grain, iron, and zinc level has allowed the development of biofortified wheat through classical breeding approach. So it is always very important if you have kind of variations in the genotypes, because it gives us an opportunity to enhance and to transfer those characters from one genotype or from one line to another line. Because we have a variation for yield, some varieties may have more yield and some may have less yield. Then we have a variation for iron concentration. Some may have more iron concentration in grain, some may have less concentration in grain. The same way zinc. So when we have such kind of variation, we can utilize those variations for classical breeding. The modern wheat cultivars have high yield potentials but are low in iron and zinc content. I will give only one example of zinc in wheat. If we see the chronological map, this starts from 1950. You can see here that we have varieties which high zinc concentration, and then there was a green revolution. We enhanced the yield, but the concentration is gradually decreasing. So till 20 10, so it was much reduced. And then we got new varieties which has been developed. So that is Zincol in Pakistan, then Zinc Shakti in India, and then there's Mile in India. So they have now the higher concentration because uh, that has been developed by special breeding for zinc only. But of course, they have also a sufficient yield potential because with low yield, you cannot commercialize those varieties. You can see here the yield. Uh, the yield of these varieties is also at higher level. So we can see from 1940 even 1952. So there is an increase in yield and there is a decrease in zinc concentration. But fortunately, what we have the biofortified varieties here, so they have also high yield. So it means they are commercializable and they are being commercialized. So Zincol, I will take the example of Pakistan. So this is the first high zinc wheat in Pakistan with plus 9 ppm zinc and with initial seed multiplication in 2016 to 17. So it has been developed and it is in the field and it has a high concentration. If we just look at the grain yield here per part, this is a part study. You can see here the grain yield is almost similar to that of Faisalabad 2008 at high zinc levels. So that is 8 milligram zinc per kg soil. But if you look at the zinc concentration or grain zinc concentration, so that is much higher than that of Faisalabad 28. It is not only when you apply the zinc concentration, but even with zero applied zinc, you are even getting more zinc content or concentration in the grain. So it means it is really biofortified or zinc biofortified uh, variety, and it's well accepted. So now the very comprehensive study is being made on this. This I have taken a protocol published by Nicola et al. They are working on Zincol 16 in Pakistan. And so they have chosen different families and they are providing that variety or the floor of that grain to those families and they are being fed on this variety floor and then they will check different parameters from that. The study has been completed partially, but the results have not been published. I have personally discussed with them, so they have some good results and also there are some confusing results as well. So that's why they have expanded this study throughout the country. Now I think more than 125 places at more than 125 places is being cultivated and then will be provided to the families and then they will get the benefit on the health of the people. So what parameter they will select or they have selected? So we know that the zinc has a beneficial effect on the immune system. The second messenger, uh, zinc signaling, hormone establishment, DNA, RNA synthesis, night vision, wound healing. So they have some parameters like night vision and they will also check about the DNA degradation and maybe some hormone metabolism. So they will check about that, and then hopefully in next few years, so we will get the very comprehensive results about that. That what is the benefit of this zinc biofortified wheat variety on actual human population? Then we have a quantitative trait loci uh, for grain, iron, and zinc. And related protein content which has been identified in the or which have been identified in the wild relatives and recombinant inbreed lines of wheat through markers distributed. So we have cuticle as cutials as well, and we have these markers to uh, uh, to biofortify our crops for high zinc and iron content. So what is the procedure of that? So we have old land races, as we have discussed in earlier, that they had low yield, but they have high zinc and iron content. So there is a QTL mapping, and if you QTL mapping, and there is another option, GWAS, that is overall association mapping. And then when these QTLs, so they have been marked, then there is a validation of candidate genes, and then these genes are overexpressed uh, through transgenic approach as well in the uh, required are in the cultivated wheat varieties which have high yield. So it means we have already high yield, and then we will have also high iron concentration, or if we are taking the QTLs for zinc, so we may have a zinc concentration. So this is how the QTLs are selected. We have, for example, here the parents. This is for iron. The blue color is for iron. So they have parents, and there is also offspring. So if we have the high grain zinc concentration, grain F iron and zinc concentration, so we have that 57 to 59. So it means we have only few here. But if we look at here that 39 to 41, so we have a high concentration. So it means we have some lines uh, which have this much concentration, and there is a high dominancy of that. So this portion is actually very important, but it is very different from the parents. If you look at the parents, P2 and P1, this blue color, and the same way the zinc, we have also the zinc with the high content, relatively high content as compared to that of parents, especially if we look at P2 and P1. So it means by this way we can have some inbred lines, and then we can have the QTLs mapping. So this is a chromosome, and there are different markers based on the QTLs that are, and they are zinc, iron, and there are so many. Even the markers are being increased day by day when people they are studying on that, because you have to mark the zone where that actually character is being controlled or that. So we are not going in detail. So there is not a mandate of this course. Plus, of course, we must know when we talk about biofortification and genetic biofortification that what is actually behind whole story. So by using that, so there have been selected different transporters or regulators which enhance the uptake of these iron and zinc, and then there is also some translocators, and then there are also some genes which are involved in the storage in the endosperm of the feed. So in this way, by transforming these genes, the yield and the grain zinc concentration or iron concentrations is being enhanced. But I don't think we have any such variety till now for wheat which has the basis of this high zinc concentration or iron concentration through transgenic approach. But there are studies. There is no variety developed, but there are studies which are, of course, in the labs and the greenhouse waiting for other procedures. So it is possible. Theoretically, it is possible, but it's not still in at the mature stages that we can say that the varieties are there. So now we could talk about the biofortification of rice with iron and zinc. We know that there are different transporters and regulators and genes has been identified for rice, and has been increased. For example, we have here various transporters. Uh, for example, one overexpression of barley, nicotinamide, 4, 2, 3. So they all are basically, they produce more mugenic acid, which enhance the Fe3 iron uptake. We know that the most of the iron in the soil that is in FEC form, so the plant cannot take up that. So these mugenic acid production basically increase the iron uptake in the form of FE3. So if when there is the iron uptake, of course, then we need also some transportation. There are also some genes which transport iron from soil to root and then root to other parts of the plant. And then we have also some genes which uh, uh, restrict the accumulation of iron into the vacuole of shoot. So it will help to translocate those iron molecules towards the grains, because when they are stored in the vacuoles, so then there will be more storage in the shoot. So we don't need storage in the shoot, we need in the grain. So once they are transported to grains, then we have also some endospermic specific expressions of ferritin proteins, and so these basically accumulate or sequester the iron in the endosperm. So we have the whole story completed in rice, and we have different expressions here which are being used to develop the iron and zinc efficient varieties. For example, iron biofortification, there is the ferritin uh, genes involvement that enhances the 7 microgram per gram derived iron. Then overexpression of soybean ferritin, then that gave us 38.1 microgram iron. Then the facilus ferritin 22.07 microgram. Then OSNAS, the nicotinamide synthase. So that gave us 19 microgram per gram driver. So these are only few examples which I have quoted here and which are being worked for rice. And so they have found that in the conventional, so they found 23 microgram dry weight iron in the endosperm. So this zone is basically endosperm, and here you can see there's more green part. It means there's more iron content. So 64 microgram per gram dry weight has been achieved by involvement of only this one. Then we have a zinc biofortification of rice. Here you can also see that there is one gene involvement which gave us 35 microgram per gram. Then soybean ferritin, so that has also enhanced the iron content, 35 microgram. And then we have OSNS2, which gave us 64 point. Actually, this is not 64. I think it's more than that. It's around about 70. So I think I have misquoted here. So anyhow, you can find from the Johnson et al. This is an open access article. You can read it from here. So the purpose to show all these that we have a very successful stories of the biofortification of zinc and iron in rice. So this is another example, rice endosperm iron biofortification you can see here. So there is a high concentration. For example, if you see here the mineral content in polished rice, and then the mineral content in the brown rice. You know, so here we can observe that in the polished we have relatively higher content, but if you look at in the brown rice we have relatively higher, and in the polished that has been decreased because the concentration is normally more on the skin of the leaf of the grain. So when you polish it, you remove it. But even then, the endospermic concentration is also in. So it means the involvement of these genes which are specific for endospermic concentration. So they work and they got high concentration. So that was all about zinc and iron. We'll talk about the golden rice, which is very famous since many years, delayed since many years, and just very political as well. Uh, we will discuss a little bit about that. How does it happen, and what is its status now, and what are the perspectives? If we look at the map, so we have severe deficiencies, are the clinical deficiencies in Africa and in India, and even we have in Yemen as well, and there are some other parts here. And in Pakistan, we have also severe subclinical. It's still not clinical, but it's a subclinical, but that is severe. Okay. So it means there is a vitamin A deficiency throughout the world if you look at the map. So there is a need to add up vitamin A in the human nutrition. So golden rice is basically rice enriched in beta carotene or provitamin. We also say provitamin A because beta carotene is it's not a vitamin A. It's provitamin A. It's a precursor for vitamin A, which is being produced in the human body. The paradigm for the international biofortification efforts. Both alpha carotene and beta carotene are particularly regarded as precursor of vitamin A. Common orange, yellow, and green colored fruits and vegetables such as carrot, pumpkin, apricots, sweet potato, and beans are rich source of these carotenoids. So the colorful, because it's basically a pigment which is in the fruits which are colorful. So golden rice is a nutritional source of vitamin A, and the vitamin A improves human immune response to them. So nowadays the era of coronavirus, and we need very strong immune system to combat the COVID-19. Uh, Photosynthetic organisms synthesize carotenoids to protect the photosynthetic apparatus from light overload. So this is the natural phenomena in the plants. Uh, Carotenoids and xanthophylls are lipid pigments that accumulate in chloroplast generally to save the chlorophyll content. That also have chlorophyll, and chromoplast that don't have. Chromoplast is in the roots and the chloroplast in the leaves. In both the carotenoids are there and give yellow and red color to tomatoes, marigolds, carrots. Chloroplast and chromoplast are convertible. Example, when tomato becomes red, the chlorophyll disappears and carotenoids are synthesized. So it means tomato is also a rich source of these carotenoids. The other crops are fruits. The mango is this rich source. This is microgram per 100 gram. Papaya, carrot is a very rich source. We should eat carrots. And then dark green leafy vegetables, red palm oil, eggs, milk, liver, cod liver oils. These are the fish oils. So they are rich in vitamin A. We can include them in our food, but if we can't, you can observe from here that uh, the mango, luckily in our country we have a surplus mango in the seasons. The people they can eat, even the poor people they can eat. Carrots are very cheaper, but the habits are not there. People they are not very habitual of eating carrots, especially the uncooked, which is very pity, and which should be because the carrot is not a much expensive vegetable in Pakistan. But people even they are not habitual of eating it, so that should be promoted. Of course, you can promote in your homes at. So there are also other liver and the cod liver also they are being also used as a rich source of vitamin. So how does it happen? So generally the plants so they have this uh, granulyl diphosphate. So they are in this form. So these are the white basically rice. So they have this molecule. So they are not converted to beta carotene. So there are 3-4 steps here. So from conversion from this to this, and there are a lot of enzymes which are involved in that. And uh, even it's a very simple way. If you look at here, so this is basically these are the steps which are involved. And what happened in case of golden rice? So the phytene lycopene, this is phytene and this is lycopene. So there are four steps here. So these four steps has been reduced by involvement of a bacterial phytin esterase, or there are two different plant esterase, phytene esterase and zeta carotene esterase. So as you have observed in the previous slides, so we have phytene esterase and beta esterase. So these are from the plant source, and these have uh, reduced these four steps to two steps. But if you use only the bacterial phytene esterase, so that will directly convert phytene into the lycopene. So in both ways it can happen. So we'll get the beta carotene, of course. Of course, there is also alpha carotene, but that is not much important when we talk about its characteristic as provitamin. But we have a high concentration of beta carotene when we involve these genes in that. Bioavailability of beta carotene is then another issue. Bioavailability relates to the fraction of carotene that the body can extract out of given foodstuff and use to make vitamin A. So because the beta carotene is a precursor and the body has to prepare it, the vitamin A from this precursor. So we know that the bioavailability, we have discussed in detail that if it's not bioavailable, so it is of no use. It is determined to a great extent by the food matrix, how within plant tissue carotenoids being stored. So it is that in which form is in the plant tissue. For example, we have protein bound, as well we are protein associated. We have also free membrane in chromoplast, and then we have crystals as in tomato and carrot. So this is probably the least available form. So we have seen that in tomato and carrot, we have a high concentration of beta carotene, but they are not all available. Even then, we have a sufficient amount in carrot, but we have seen the 2000 microgram in carrot, but all that is not available to the to the animals or humans, because the humans digestive system cannot extract that to make it vitamin A. So vitamin A requirement of is 900 to 700 to 900 microgram per day. This is roughly estimated for a normal body. So this makes a difference. So when you added some genes from daffodils, and when you added some from maize, because the maize has already these beta carotene in maize. So there was intervention from daffodils and from maize, and we have observed here, our researchers, they found that you can increase 1.6 to 35 microgram beta carotene per gram in the rice. So the white rice has almost no beta carotene in that. So, and this color development also tells us about the concentration. If it's more brown, it will have more carotene as compared to less golden. So this is a golden rice which has been produced with the high vitamin concentration. But so there are also critical analysis. There is a lot of critique on the golden rice. There is no other crop in the world, transgenic crop, which has faced a lot of criticism. This is almost recent, I think 2017 or 18 study. So the IRI, International Rice Research Institute in Philippines, so they found that low level of beta carotene is 1.2 against claimed 1.6. So 1.6 was considered that will fulfill the requirement, but they have found at their farm, so that was 1.26. So that was the one reason that the whatever that has been claimed that has not been achieved at Erie Farms. Then the rapid degradation of beta carotene during the storage, because the rice is mostly stored. So they have also found that there is a degradation of beta carotene. It means when it will be utilized, there will not be sufficient amount of beta carotene in that. Higher level of beta carotene has been reported from plant grown in greenhouse, not in the field. So they have also gave the reason why this concentration was low, because the people who has reported, so they grow their plants in the greenhouse. So that may be the reason that they have found 1.6 to even more concentration in that, but that was not achievable in the field as per Erie experiments. Then efficacy studies have not been conducted for targeted population, because we have a targeted population in different areas where there is a vitamin A deficiency, and how much rice they need to fulfill all these requirements of vitamin A. For example, if we have a low concentration, so we cannot give the people to eat more rice. It means we have to fit the concentration or the required amount in the rice which has been being used uh, by the people. Uh, but that was the critics from Erie. There are many other studies who are supporting this. Herald did as a genetically, so this is published in Science Daily, genetically modified crop with the potential to save millions of lives. Golden rice has just been approved as safe for human and animal consumption by regulators in the Philippines. In the Philippines, again, there was another study where they found that it is safe. So there is no issue. The rice is a beta carotene enriched crop that is intended to reduce vitamin A deficiency, a health problem in very poor areas. But a new study finds that most families at risk for vitamin A deficiency can't grow golden rice themselves, and most commercial farmers won't grow it right. So the people who are deficient, so they might not have their own land, so they cannot grow for themselves. And the commercial farmers, so they are not growing that golden rice due to different reasons. So it means we are not getting golden rice into our population. So these are the two articles which are very important, and I suppose that you should read it, because this is important really. When we talk about the global health, we should focus on such type of articles. So with that, I would like to thank you very much, and we will have then next biofortification lecture, the remaining crops. Thank you.